All right, let's now talk about compounds that have two or more chiral centers. Up until now, we've been talking about ones that only have one. When you have two or more chiral centers, not only do you have enantiomers, but you have another type of stereoisomer that we're going to talk about. Okay, so a compound with one chiral center has two stereoisomers, a pair of enantiomers. A compound with two or more chiral centers has four possible isomers. Okay, so this is an amino acid, threonine, has two chiral centers. You can see one right here. And notice it's in a Fisher projection. So which groups are going to the back? It's the top ones. This is and the bottom. This is going to the back. This is going to the back. These are both coming out towards me. So if I were to draw this with the wedges and dotted lines, just to remind you, okay, this is kind of how you'd write it. Now it's different because we have two chiral centers this time, but the ones in the vertical are always going back, and the ones in the horizontal, sorry about the writing, I lost the pen for the little pad that I use, so I'm trying to draw this with a mouse, and you can see it's really not that great, but I think you get the point of view, or the point. All right, the NH and the H are coming out towards you, they're wedged. The H and the OH is wedged, and these two in the very top and the bottom are going back in the plane. That's a Fisher projection. Okay, so I have two chiral centers. Okay, so I have the enantiomer. So here's one. Let's kind of analyze this. The carboxylic acid, carboxylic acid. This H, shine it in the mirror. That's the first thing I see from the mirror, and the NH2, which is in the back, is in the back. And then the OH here is on the right side. Here the OH is on the left side. These are enantiomers. Okay, mirror images. Now, these are two different compounds, enantiomers. Now, what if I were just to switch one enantiomer but keep the other one exactly the same? That would be this compound. So notice these, okay, so I'm comparing A and C. Okay, A and C are a little different. Okay, notice this carbon, the H and the OH is identical. The H and the OH, same side. OH on the right, OH on the right. But this is switched. NH2 is on the right, NH2 is on the left. So it's not a mirror image. Notice these are not mirror image. I'm going to put MI for mirror image, not mirror images. Okay, but they are still the same compound, right? It's the exact same compound. It's still 4 carbons, and you have the NH2 on the second one, and the OH on the third one, and carboxylic acid. But in 3 dimensions, they are different. So they are not mirror images, but they are still stereoisomers of each other, meaning they are isomers stereoisomers, meaning it's the same compound, but they differ in 3 dimensions. Okay, so that has a different name. Okay, notice this compound, same thing. All right, it's not a mirror image. This group has switched this time, but this group is the same. Okay, all right, so these are enantiomers. These are mirror image of each other. If you look at these two compounds, these are also mirror images. Okay, however, this group of compound is not the mirror image of these. Only one of the stereocenters has changed. So these are what's called diastereomers. Okay, and people sometimes get these confused, diastereomers, but basically you have to remember, if every stereocenter is completely changed, if every chiral center has switched and it's a mirror image, it's an enantiomer. Okay, if one of the centers is a mirror image but the other one is identical, then it's a diastereomer. Okay, and you'll see on the same slide. So A and B and C and D are enantiomers, but A and C and B and D are what's called diastereomers. Okay, actually it's A, C, A and D are diastereomers. B and C and B and D are diastereomers. Okay, so you've got enantiomers, which are mirror images, not superimposable. Diastereomers are non-mirror images, so obviously they're not superimposable, but they are still the same compound. Okay? We will definitely talk more about this in class because people sometimes, they understand enantiomers, but when you start talking about diastereomers, things get a little confusing. But if you watch this and if you really look close and analyze these compounds, you have to look close at these and analyze them. Otherwise, I don't want you just to kind of sit there and say, oh yeah, I see what you're saying. You have to actually look at it and really analyze these compounds. Okay, and your book I'm sure has more pictures in it, which are probably better. Um, Boiner. Okay, so notice this OH on the right and H, OH on the right and H. Those are identical, those stereocenters. But these are opposite. Okay, NH2 is on the right here, here NH2 is on the left. So these are the same, but these are different. So it's not a mirror image, but one of the centers are different. Therefore, it's a different molecule. Okay, so I'm sorry, it's a different stereoisomer. So they're called diastereomers. Okay, this is kind of a summary of all the isomers that we've talked about. We've talked about constitutional isomers, isomers having atoms bonded to different atoms. So like ethanol versus dimethyl ether. Okay, same formula, different structures. But then stereoisomers, basically it's the same compound, except you have a mirror image that's not superimposable, or you have one that's not a mirror image, just like what we talked about. All right, so this hopefully it summarizes everything up. Um, and we will definitely talk more about this when we get to class. We will look at models. Please, obviously, if you are watching this, and you don't have to watch it before class, but you have to watch this before class because I want to do an activity so that you can really kind of see all the stuff that's going on. All right, 15.9 is kind of a really cool way, again, kind of to show you how this works inside our bodies. It's really, so what this basically says is that it's not the functional group that our body recognizes, it's the shape of the molecule, not necessarily the functional group, which I don't know if I agree with that because you do have hydrogen bonding with carboxylic acids and alcohols, which have a different effect on our proteins and our smell receptors, but shape does have a huge impact on it, okay? And this kind of analysis, I think, is good. You have cyclooctane, okay, which is 8 carbons, just carbons and hydrogens. Then you have this molecule, which is hexachloroethane. Now, if you look at the shape of these molecules, this is what's called a space filling model. You've probably seen these in the book. This is actually what they think the atoms or the molecules look like. It's blobs of electrons. It's not obviously, a Cl and a stick and a carbon attached to it. It's blobs of electrons. Okay. Now, these two atoms or molecules smell exactly the same. Okay. Our noses smell it, and it smells like camphor. Camphor is like what's in Vicks Vapor Rub. If you have ever smelled Vicks Vapor Rub, that real intense smell that clears up your sinuses, that's what the smell of camphor is. So what this book is saying is that these compounds have the same smell as camphor. All right, these are not even close to the same compounds. I mean, this has 8 carbons and 16 hydrogens. This has 2 carbons and 6 chlorines. But because of the size of the chlorines, they have a similar shape to them. You can see that the shapes are the same. It's this big spherical ball. Well, camphor is basically all composed of carbons and hydrogens. It does have an oxygen, but it's shaped like a ball. And they think our smell has nothing to do with the atoms on it. It's basically what receptor site it's going into based on the shape. And it's triggering our brain saying, hey, that has a hot smell to it, or a camphor smell. All right, so I think that's kind of interesting. And here it's kind of trying to show you, a very easy way of how this works is you have a receptor site on the olfactory hair, which is right above our soft palate inside of our noses. Okay, and there's a binding site there that fits perfectly for octane. So here's kind of a blow up of it. Here's kind of a smaller view. Here's kind of a larger view. So as soon as you smell something in your nose, we have these little tiny hair-like nerves that are hanging out of our nose that kind of absorb everything that we smell, and it triggers a chemical response into our brains. Okay, so that's kind of what they're showing here. But they're showing that this molecule, it doesn't matter the receptor site, or I'm sorry, the functional groups, it's the shape of it. It fits inside of this particular receptor site. Okay, and that's kind of how they're saying smell works. This is very interesting. I'm going to show a video about this in class when we meet. And it's a difference between caraway seeds and spearmint leaves. Uh, Caraway is the smell of rye bread. Okay, everyone kind of knows what rye bread has, that rye smell to it. Um, that's what caraway is. Spearmint, you all know what spearmint smells like. Well, believe it or not, they are the exact same molecule. These compounds are the exact same molecule. However, they are enantiomers of each other. All right, the compound is called carvone. All right. So one enantiomer gives that rye smell. The other enantiomer gives a spearmint smell. But can you believe these are the identical compounds? They just are different by the chirality, and they are such a different smell. Would you ever think that rye bread and spearmint are related at all? I mean, I would not. I only know it because I've seen this slide probably 1,000 times, and I've seen the video I'm going to show you 1,000 times. Just based on the chirality, it has a different effect inside of our noses, and our brains trigger it and say, well, this fits in this receptor site, therefore, hey, that's the smell of rye. This compound doesn't even matter what the structure is. It fits into this receptor site, and that's that spearmint odor. Okay, so again, that's why they're saying it doesn't matter what functional group, because technically these should smell exactly the same, right? There's no difference. I mean, well, it's not that much of a difference. Well, obviously inside of our bodies, it's a huge difference. All right, again, this is why I found chemistry, specifically organic chemistry, is so fascinating. These slight different changes have such a huge, profound effect on a living organism. All right. So I'm going to end it there. I think it's kind of a cool way to end it. I honestly, guys, I really hope you're finding these lectures interesting and the material interesting. I love this book. I really like how the author is taking the stuff we're learning and putting it in real life examples. And I'm hoping you guys are learning tons of information because I find this stuff fascinating. So let's talk more about this when we meet. I'm not going to do a lecture like how I have been doing. I want to kind of make some molecules, and you guys ask questions, and we can talk about all this cool stuff. All right. So watch videos, do homework, ask questions. See you at next lab.